This is Ravindra Babu. Welcome back to my channel Silver 90s Bionet. In the last video, we were discussing about the three models of DNA replication. Those are semi-conservative model, a dispersive model, and a conservative model. Among these three models of DNA replication, the semi-conservative model of DNA replication is only having experimental proof. The remaining two, they don't have any experimental proof for DNA replication. In this part of video lesson, we are going to discuss that experimental proof for a semi-conservative model of DNA replication. The experiment is Meselson and Stahl's experiment. Here, Matthew Meselson and Franklin Stahl are the two scientists who conducted this experiment to prove DNA is undergoing semi-conservative model of replication in the year 1958. Actually, this experiment is a quantitative experimental proof. Quantitative experimental proof. I will come across later why this experiment is considered to be as a quantitative experimental proof at the end of this video lesson. And in this experiment, they worked on E. coli cells. Remember, the E. coli cell is a prokaryote. So, to prove the DNA is undergoing semi-conservative model of DNA replication, they used a E. coli cell. What is that? That is a prokaryotic cell. Keep remember this. And for their experiment, they used what they used? They used N15 heavy isotopic form of nitrogen. Okay. And they also use cesium chloride density gradient centrifuge for their experiment. The question is, why they selected this N15 heavy isotopic form of nitrogen and cesium chloride density gradient centrifuge for their experiment? The answer is, the N15 heavy isotopic form of nitrogen is actually heavier in density than the normal nitrogen. The normal nitrogen is N14 nitrogen, which is generally present in the biomolecules, especially DNA of living organisms. This N14 form of nitrogen, which is present in the living organisms, is lighter in density than N15 form of nitrogen. So, that is the reason. And come to cesium chloride density gradient centrifuge. The cesium chloride is a chemical substance which shows the similar density values to that of the normal nitrogen present in DNA. So, N14 DNA and cesium chloride shows similar density, so that they prefer the cesium chloride density gradient centrifuge for their experiment. Now, let us move on to the experimental procedure followed by Meselson and Stahl prove a DNA is undergoing semi-conservative model of replication. What they did here? First, they took E. coli cells of normal DNA. Normal DNA means a DNA having N14 form of nitrogen. Okay. To this E. coli cells of an N14 form of DNA, they are allowed to grow on a N15 medium for several generations. So, after several generations, here N15 form of nitrogen gets incorporated into the newly synthesizing DNA of the newly forming E. coli cells. So, after several generations, all the E. coli cells, whatever produced on this N15 medium, would have what type of DNA? N15 type of DNA. So, that N15 completely replaced the N14 in DNA molecules of E. coli cells. At this condition, they isolated the DNA of these E. coli cells of N15 forms Okay, and allowed for centrifugation by using cesium chloride density gradient centrifugation methods. After centrifugation, they got a band at the bottom here. Okay, here the reason is N14 DNA is heavier in density than N14 DNA or heavier in density than cesium chloride, so that this N15 form of DNA which is present in this E. coli cells grown on N15 medium form a band at the bottom. Okay, that is a result at the first step of their experiment. So they continued their experiment further here. They allowed this E. coli cells of N15 DNA to grow on N14 medium for about 20 minutes first. Here 20 minutes is a generation time for a E. coli cell. Nothing but a E. coli cell can divide to give 2 E. coli cells. It takes 20 minutes, that is the generation time. After growing this E. coli cells of N15 DNA on N14 medium for about 20 minutes, they got a first generation. This is the first generation. The 2 E. coli cells are formed. These 2 E. coli cells are having hybrid DNA as the DNA is undergoing semi-conservative model of replication. Say here, this is the pink strand is a parental DNA. This white strand is a newly synthesized DNA containing N14 nitrogen, as the E. coli cells are grown on N14 medium. Same in this E. coli cell also. The DNA is having one strand of parent type. Parent type means N15 strand, and one strand is normal type, means N14 strand, as the E. coli cells are grown on N14 medium. At this stage also, they allowed this isolated DNA of first generation E. coli cells to centrifugation by using cesium chloride density gradient centrifuge. Here they got a band at the middle. This band at the middle indicates that the DNA of the first generation of E. coli cells is hybrid. Nothing but one strand is a parental type N15 type and another strand is N14 type, and newly synthesized. Here also one strand is a parental N15 type and one strand is newly synthesized N14 type. So these are the hybrid DNAs of 2 E. coli cells of first generation, so that the band at the middle. Nothing over here, 100% of first generation are what type? Hybrid type. Keep remember this. They allow the same E. coli cells for a second generation also for another 20 minutes. Nothing but after 40 minutes they got a second generation of E. coli cells. In the second generation they got 4 E. coli cells. Among the 4 E. coli cells, the 2 E. coli cells are having what? Hybrid DNAs. Hybrid DNAs. As a replication is semi-conservative model, in these E. coli cells DNA will have what? Parental N15 type and a newly synthesized N14 type, as the E. coli cells are growing on N14 medium. And here this is the parental DNA of N14 type, and newly synthesized DNA is also N14 type, as the cells are growing on N14 medium. Like that, here in second generation, the 2 E. coli cells are having light DNAs. What are these? Light DNAs, light in density, and the 2 E. coli cells are having hybrid DNAs. Hybrid DNAs like this. Okay. Here also, what they got? 50% of hybrid DNA and 50% of lighter DNA. Here also the isolated DNA of E. coli cells of a second generation and allowed for a centrifugation in cesium chloride density gradient centrifuge. Here they got a middle band and a top band. Middle band indicates that what? The 2 E. coli cells of a second generation having hybrid DNA. This is hybrid DNA, and the top band indicates that the 2 E. coli cells are having lighter DNA. Lighter DNA. Here completely hybrid DNA. Here heavy DNA. They continued their experiment for about third generation and fourth generation also. In third generation, what happened? Third generation, it takes about 60 minutes. After 60 minutes, they got third generation. In this third generation, they got how many cells? 80 E. coli cells. Among these 80 E. coli cells, 2 E. coli cells are having what? Hybrid DNA, and remaining 6 E. coli cells are having lighter DNA. 2 means what? 25% are having hybrid DNA, and 6 means 75% are having lighter DNA. Here also the centrifugation has shown the same result, that is a middle band and a top band. In fourth generation also, fourth generation means after 80 minutes, fourth generation, here they got 16 E. coli cells. Among the 16, the 2 are having hybrid DNA and remaining 14 are having the lighter DNA, as the cells are growing on N40 medium, light medium. Okay, and among these, the 2 means what? 2 means just 12.5% are having hybrid DNA, and 14 means 87.2% of E. coli cells are having lighter DNA. In this case also, after centrifugation, they got same a middle band and a top band. Nothing but from second generation onwards, they got a middle band indicating E. coli cells of hybrid DNA, and the top band indicating the E. coli cells of lighter DNA. If you carefully observe this experiment conducted by Meselson and Stahl, here from first generation to last generation, you could observe that the decrease in hybrid DNA content. Here the hybrid DNA content first generation is 100%, in second generation is 50%, and third generation at just 25%, and in fourth generation it is just 12.5%. Here hybrid DNA means parental DNA. What is the parental DNA? The N15 DNA is a parental DNA, because the hybrid DNA consists the N15 parental DNA, so that we are considering the hybrid DNA as a parental DNA. So that decrease in hybrid DNA from first generation to last generation means decrease in a parental DNA concentration. Okay. So based on the decrease in the quantity of parental DNA from first generation to last generation, here we are saying that this experiment is a quantitative experimental proof for the semi-conservative model of DNA replication. And one more point we have to observe in this experiment is, from second generation onwards, they got a middle band and a top band. In third generation also they got middle band and top band. In fourth generation also they got middle band and a top band. This result of centrifugation also indicates that DNA is undergoing a semi-conservative model of replication. Here you may got one doubt. Just based on the decrease in the quantity of parental DNA from first generation to last generation, and just based on a formation of a middle band and a top band from second generation onwards, how can you say that the DNA is undergoing semi-conservative model of replication? Let me clear this. Say if DNA is undergoing a conservative model of replication, DNA replication, if it is conservative, what type of result Meselson and Stahl could got in their experiment? Let us see that. If you observe the first generation of Meselson and Stahl's experiment, DNA being showing a semi-conservative model of replication, we got 100% hybrid E. coli cells having hybrid DNA molecules. So that the result of centrifugation is middle band representing this hybrid DNAs of two E. coli cells. If the DNA is undergoing conservative, let us see what type of result they could got here. So in first generation, one E. coli cell could have completely a parental DNA, that is N15 DNA, and another E. coli cell could have completely a newly synthesized DNA, that is N14 DNA. So the result of centrifugation could be like this. The N14 DNA containing E. coli cells forms a band at the top, and the N15 DNA containing E. coli cells forms a band at the bottom. So this is heavier N15 DNA and this is lighter N14 DNA of the E. coli cells of first generation, but actually they could not get this result, they got this result in their experiment. By this they could able to disprove that DNA is undergoing a conservative model of replication, but DNA is undergoing a semi-conservative model of replication. And in the second case, that is a DNA replication is if it is dispersive. What type of result Meselson and Stahl could got in their experiment? So in this case, if the DNA replication is dispersive, one E. coli cell could have patches of N15 and N14 DNAs like this, and another E. coli cells also could have the patches of N15 and N14 DNAs like this. So that the result of centrifugation in this situation will give a middle band like this. Here the middle band indicates what? N15 and N14 hybrid DNA, but the difference is in semi-conservative model, the two E. coli cells are having N14 and N15 strands, so that is a hybrid DNA. But here the situation is different. The DNA of two E. coli cells are having the patches of N14 and N15 DNAs. So in semi-conservative and dispersive, they got a middle band in the first generation. So in order to disprove that DNA is undergoing a dispersive model of replication, here they continued their experiment further. So after 40 minutes in the second generation, here also they got only the patches of N15 and N14 DNA in all 4 E. coli cells of a second generation. So the result of centrifugation here is same as of first generation. Here also they got only a middle band representing hybrid DNA of N15 and N14. As DNA is undergoing semi-conservative model of replication, they could got this type of result and they could not get this type of result. So that by this also they came to conclusion that DNA is undergoing a semi-conservative model of replication, not through dispersive and not through conservative. And finally, the conclusion is, if the DNA is undergoing a semi-conservative model of replication, the result of centrifugation will be like this. In first generation, you will get a middle band. In second generation, you will get a middle band and a top band. If the DNA replication is conservative, the result of centrifugation at first generation onwards will be a bottom band and a top band. And if the DNA replication is dispersive, from first generation onwards, we will get only middle bands. Thanks for watching this video. Please like, share and subscribe my channel.